Okay, good morning, everyone, or good evening if you're in China. It's a great pleasure to welcome you to this month SCBA-NIH Member Trafficking webinar. So I'm Yihong Ye, Senior Investigator at NIDDK, and it's a great pleasure to introduce today's seminar speaker, Dr. Xiaochen Wang, a chair professor at the Southern University of Science Technology in Shenzhen, China. So Xiaochen got her bachelor degrees from Shandong University, and then she did her PhD in Dr. Chen Zhangliang's lab in Peking University from 1996 to from 1994 to 1999, and then she moved to the United States and did her postdoc work with Dr. Jing Xue at University of Colorado. Where she started to use silicons to dissect the mechanisms or cellular mechanisms underlying cellular apoptosis, which took her to the field of lysosome. And she established her own independent research group in 2006 in the newly founded National Institute of Biological Science in Beijing. And within 5 years, she was promoted to associate investigator. And in 2016, she moved to the Institute of Biophysics in Beijing, became a principal investigator there. And then more recently, she got recruited to the Southern University of Science and Technology in Shenzhen as a chair professor. And through her career, Xiaochen made a lot of groundbreaking discoveries in the field of lysosome biology and membrane trafficking. And she received numerous awards, including HHMI Young Investigator, Early Career International Scholar Award, and the Distinguished Women Scientist Award in China. And she's currently serving as the editorial board member of the Journal of Cell Biology. And so I knew her work from many years ago by reading her papers, but only got to met her recently in a meeting in Shenzhen. And when I visit her labs and talked to her, I was really infected by her passion to science and also very inspired by her beautiful works that linking detailed cellular mechanisms of membrane dynamics and lysosome functions all the way to animal development. So I thought that I have to invite them to give a talk and to this community. So it's great pleasures that Xiaochen accept invitations and can spend her time with us. So Xiaochen, the podium is yours. Thank you.
Thanks, Yihong. Can you hear me?
Yes.
Yeah, thank you. Thank Yihong for this invitation, the introduction and the invitation. I'm very happy and appreciate the opportunity to share our work with scientists from both China and the US. Although I couldn't see you guys in face, but I hope that I can keep you in this talk by our work. As Yihong introduced, my lab is interested in lysosome-mediated cellular degradation. As you probably all know, that lysosomes are the centers of degradation and the recycle. It receives the cargoes derived from endocytosis, phagocytosis, and autophagy, and recycled in resulting catabolized. It also serves as a signaling hub that can detect the intracellular and extracellular changes and coordinate the cellular responses, and therefore to maintain the cell homeostasis. Lysosome dysfunction is associated with various human diseases, including about 70 lysosome storage disease, autoimmune disease, tumor genesis, and neurodegenerative diseases, indicating the importance of lysosome in physiology. Although the disease relevance of lysosome dysfunction is quite clear, however, we know very little about the pathogenesis of lysosome-related diseases, and largely due to the complexity of lysosome-involved cellular processes. So one motivation that we study lysosome is to dissect its regulation and the function using a multicellular model. And we hope that by doing so, we can contribute to our understanding of the pathogenesis of lysosome-related diseases. So as Yihong just introduced, that our early work actually focused on the clearance of apoptotic cells, which are delivered to lysosomes through phagocytosis. The apoptotic cell clearance is the integral part of the whole cell death program. Defects in apoptotic cell clearance not only impairs execution of death, and also it could cause autoimmune response and persisting inflammation because the toxic content may be releasing from the uncleared apoptotic cells. So we use C. elegans as a model to dissect the apoptotic cell clearance because worm has many developmental cell deaths. For example, it has 131 somatic cells die during embryonic and larval development, while 300 to 500 germ cells die during oogenesis. And these apoptotic cells, as you will see in this movie again, they look like a button pointed by this arrowheads. And we can directly observe these button-like apoptotic cells under microscope so that we can follow their appearance and clearance. Then we performed genetic screens to identify genes that act at a different step of apoptotic clearance, including recognition, engulfment, phagosome maturation, and cell corpus degradation. We have identified the lipid binding protein and the lipid transferring protein that mediates PS signaling important for the recognition of apoptotic cells. And we found that the phosphoinositol cascade that regulates both engulfment and phagosome maturation. We also identified multiple RAP-GTPase that act coordinatedly to regulate phagosome maturation and the cell corpus degradation. And later on, we switched our research direction from phagocytosis to lysosome for several reasons. And one direct reason is because of this mutant QS42. We identified this mutant through a cell death-related screen because we thought at the beginning that this mutant contained many cell corpses as pointed by these two arrows. And later on, we found that these structures, the corpse-like structures, can be well stained by lysotracker red, suggesting that they may be enlarged lysosomes. And indeed, in wild-type cells, the lysotracker labeled very small vesicles, and these are normal lysosomes, while in this mutant lysosome are significantly enlarged. And as shown in this image, you can see the lysosome, the enlarged lysosome is equal to the nuclei in size. And the overall volume of lysosome in this mutant is significantly larger than in wild type. And then we found, in addition to lysosome enlargement, QS42 mutant also affects lysosome cargo degradation. We first examined the degradation of apoptotic cells because we studied phagocytosis at that time. Here we use RAP7 to label phagolysosome, and we use histone to label apoptotic cells. As you can see in wild type, the histone GAP is degraded quickly within the phagolysosome. However, in this mutant, the histone GAP persists for quite a long time, indicates that apoptotic cell degradation is impaired in this mutant. And also we saw accumulation of high levels of UC protein, which is an endocytic cargo that delivered to lysosome for degradation during embryogenesis. And we saw accumulation of SQST1, which is a P62 homolog in worms, suggesting that autophagic cargo is also accumulated. So this mutant accumulates accumulates endocytic, phagocytic, and autophagic cargos in lysosomes. And we then cloned affected genes and found that it encodes a putative lysosome membrane protein belonging to a lysosome amino acid transporter family. And the founding member of this family is the cystinosin, which is a lysosome cystine transporter, while CTS1 is the worm homolog of cystinosin, and this is the LAT1. So we purify the lysosome from worms and determine the amino acid concentration in lysosomes. We can see that the CTS1 mutant, the lysosome contain high levels of cystine, which is consistent with the function as a lysosome cystine transporter, while the mutant, the QS42 mutant, does not accumulate cystine, but contain high levels of lysine and arginine. So we suspect that this mutant actually, the affected genes is a lysosome lysine and arginine transporter, and we therefore named this gene as LAT1, which after a lysosome amino acid transporter, shorter for lysosome amino acid transporter. To determine whether it is indeed amino acid transporter, we adopted a cell-based amino acid transport assay. In this assay, we removed the lysosome targeting sequence of LAT1 and its human homolog PQLC2, so that these two proteins can be targeted to the plasma membrane. And the topology is similar as that on the lysosome surface. Then we can test the amino acid transporter assay using a cell uptake assay. And by this cell-based assay, we found that LAT1 and the PQLC2 exhibit very well transporter activity towards both lysine and arginine, while a single amino acid mutation totally abolished the transporter activity. So our work suggests that the LAT1 is a lysosome lysine and arginine transporter. It localized to lysosome membrane and transport lysine and arginine from lysosome lumen to the cytosol. So and in this way, supporting the protein synthesis, which is important for embryonic development. While in the LAT1 mutant, lysine and arginine are trapped in the lysosome lumen, which caused cytosolic deprivation of these two amino acids, and this triggered the AAR, which is the amino acid response pathway, leading to repression of global protein synthesis and ultimately causing the retardation of embryonic development. As I have mentioned earlier, the cystinosin is the founding member of the lysosome amino acid transporter, which LAT1 belongs to. Cystinosin is a lysosome cystine transporter, and its loss function causes accumulation of free cystine in lysosomes, and this leads to a lysosome storage disease, cystinosis, that affects multiple organs, including central nervous system. The most effective therapeutic agent for the treatment of cystinosis is cystimine. It is thought that cystimine can convert free cystine in lysosomes to cystine and a lysine analog, which is the mixed disulfide of cystine and cystimine. And it is thought that this lysine analog can be transported out of lysosomes through a lysine transporter, and therefore cystimine can deplete the free cystine in lysosomes. And therefore, it can be used in treatment of this disease. However, because the putative lysosome transporter has not been identified, so this stays as a hypothesis. Now we have a lysine and the arginine transporter in hands. We then decided to test this hypothesis in worms. The CTS1 mutant, which is the worm cystinosin, this mutant accumulated huge lysosome in celomocytes. This is a macrophage-like cells in worms. And then we use the cystimine treatment. We can see that the cystimine treatment significantly reduced the huge lysosome phenotype in CTS1 mutant, suggesting that in worms, the cysteamine can work like in patients. It probably can deplete the free cysteine and therefore suppress the huge lysosome phenotype. Well, more importantly, we found that the huge lysosome still present in the LAT1, the CTS1 double mutant, which suggests that LAT1 is required for cysteamine treatment, the suppression effect of cysteamine. And to further test that, we again purified the lysosome from worms and determine the cysteine levels in lysosomes. Consistent with the huge lysosome phenotype that CTS1 mutant contain high levels of cysteine in lysosome, while the LAT1 CTS1 double mutant also accumulate cysteine in lysosomes. Well, after cysteamine treatment, the cysteine levels reduced significantly in CTS1 single mutant, but still remain at high levels in LAT1, the CTS1 double mutant. So this data further support that LAT1, probably at human PQLC2 as well, can mediate the depletion of cysteine in lysosome by cysteamine, and therefore provides a molecular explanation of how cysteamine can elevate a lysosome storage disease. The LAT1 work actually triggers our interest in lysosome and promoted us to look at the lysosome more closely. And surprisingly, we found that C. elegans lysosomes are highly dynamic. We see both vesicular and tubular structures, and we saw that these lysosomes are constantly moving and converting between these two morphologies. The lysosome dynamics exhibit a clear cell type specificity and is associated with development, aging, and stress conditions. So we are really curious about the cues that can trigger these dynamic changes of lysosomes and the cellular processes that involve these lysosomal changes. We wonder how they are regulated and whether they have any physiological significance. So to address these questions, we developed tools in worms to label and trace lysosome in live C. elegans under different conditions. And also we performed genetic screening to isolate mutants with abnormal lysosome morphologies or mutants that accumulate high levels of GAP-glytin-3, which is a biosensor for membrane damage, so that we know that the affected genes is probably involved in regulating lysosome integrity. We then clone the affected genes and study their function. From the morphological screen, we have identified two types of the mutant. We call them Lima mutant as a lysosome morphology abnormal. One type of these Lima mutants accumulate significantly enlarged globular lysosome, but lacks the tubular structures. We call them Lima E. While another types of Lima mutants accumulate extensive tubular lysosomes. We call this mutant Lima T. And we found that the Lima E mutant affects various lysosome genes, including this lysine and arginine transporter LAT1, which we have previously identified from cell death-related screen. And also we found this Lima E mutant affects lysosome lipase, endoribonuclease, calcium channel, and cathepsins. And the mutations in these genes affect lysosome morphology and function, and they all exhibit obvious defects in embryogenesis. C. elegans embryos are covered by tough and impermeable eggshell, so all of the nutrients that are required for embryogenesis derive from degradation of macromolecules that pre-store in the oocytes. Our studies suggest that the lysosomes digest the macromolecules and recycle the recycling catabolites, and so that provides nutrient for embryogenesis. So to give you an example, the QS245 and the QS348 mutants affect RNST2, which is the RNST2 family of indorep nucleus, and QS348 mutant affect lysosome phospholipase A2. In both mutant accumulate significantly larger lysosome as in the LAT1 mutants. And our work suggested that the macromolecules that pre-stored in oocyte and embryos are delivered to lysosomes through autophagy and endocytosis. And the lysosome phospholipase A2, the LPIO2, act first to disrupt the vesicle membrane so that cargo can be released from the vesicle, while the lysosome hydrolysis, such as RNC2, it digests the ribonucleus and the resulting nucleotide are recycled to the cytosol to maintain the nucleotide homeostasis, while the lysosome transporters, such as LAT1, export lysine and arginine produced by protein digestion to maintain the amino acid homeostasis. And in this way, the lysosomal proteins coordinate to maintain cell homeostasis and provide nutrients for both embryonic and level development. So C. elegans, after embryogenesis, worms undergo a 4 consecutive level stage before entering the adult stage. At each end of a level development, worms undergo a molting stage when they shed and resynthesis their cuticle so that it can accommodate the growth, the level growth. And very interestingly, we found that the lysosome morphology changes specifically during molting stage. As you can see here, at the intermodal stage, lysosome are most vascular, while a large number of tubular lysosomes formed during molting stage. So let's look at the C. elegans molting more closely. Worms has a collagenous cuticle, which is anchored at the apical side of the epidermal cell called hyper7. And these cells is big syncytial cells that contain 139 nuclei and very big that can cover the whole mid-body of the worm. And when worms enter the molting state, the first thing it needs to do is to detach the old cuticle from the underlying hypodermis. And then the new cuticle component are synthesized and secreted from the underlying epidermis, the hyper7 cells, to produce a new cuticle. And then the new cuticle is re-anchored with the hyper7. And worm is escaped from the old cuticle to complete the molting cycle. And interestingly, worm is at the sleep-like stage when the old cuticle is detached and when the new one is being synthesized. It takes about 2 to 3 hours. And then the sleep-like meaning that worms do not move, do not eat. And after that, it wakes up to resume the feeding and motility so that it can escape from the old one. And the morphological change of lysosomes occurs specifically during this sleep-like stage, while after the new cuticle is resynthesized and anchored. When worms wake up, the lysosome resume mostly the vesicular morphology. And these lysosomal changes occurs specifically in the hyper7 cell that connected with the cuticle. In addition to the morphological change, we can see here the lysosome tubule. We can not only see by using the fluorescence microscope, but also we can see under EM these long tubular structures with membrane layers and intraluminal vesicles inside of the tubules. In addition to that, we also found that the lysosome biogenesis dynamics, maturation, and the degradation activity all increase at molt, indicating that the lysosomes are activated specifically at the molting stage. To show you an example here, we use the GAP and the cherry fusion protein to detect the acidity or the maturation rates of lysosome. As you can see here in the intermode stage, we can see both GAP and the cherry suggesting that it is still at nutrient acid. It's not fully acidified. While at the molting stage, the GAP is quenched. Well, we can only see the cherry fluorescence showing a tubular structure. And we we take different time points and measure the co-presence of GAP and the cherry. We can see that at the molting stage, the GAP fluorescence disappeared much faster than that in the intermode stage and the post-molting stage, suggesting the maturation, lysosomal maturation is accelerated at molting. Our data suggests that the lysosome is activated at the molt to facilitate the degradation of old cuticle component, while the resulting catabolites are used to build a new cuticle so that the cuticle replacement is facilitated. Then we want to know how our lysosome activated specifically at the molting stage and in hypodermis, because it only takes 2 to 3 hours. Then we focus on the anchoring of cuticle with hypodermis. As I have mentioned earlier, that cuticle is anchored with the HEP7 cells at the apical side, and this attachment is mediated by celiac and transepidermal hemidesmosomes. The apical CE-HD anchors the cuticle with epidermis, while the basal HD connect the basal side of epidermis with basement membrane, which connected with muscle. And when worms enter the molting stage, the old cuticle need to be detached from the underlying epidermis, and so we wonder whether the disruption of this attachment may serve as a signal to activate lysosome. We tested this idea first by examining the level of apical HT levels during molting. As we can see, the intensity of the MAL4, which is a co-component of the apical HD, decreased significantly at the molting stage, as well as the intermediate filament, while the basal HD component does not change much. And this is consistent with our hypothesis that the attachment structure between cuticle and epidermis may be disintegrated at the molt. Then we further tested by knocking down this apical HD component MAL4, and we can see that the MAL4 RNA leads to the appearance of tubular lysosomal structures even in the at the intermold stage. And also GAP fluorescence disappeared while cherry exhibited a tubular structure in MAL4 RNA worms at the intermode stage. So this indicates that disruption of the cuticle epidermis attachment can lead to lysosome activation. We performed a qPCR analysis and found that many of the VHA genes, which encodes VATPase subunits responsible for the acidification of lysosomes, is upregulated specifically at the molt, and all of these VHA VHA genes and then pointed by this red arrow, we can see it is upregulated at the molting stage, this pink one, while the expression level reduced post-molt. So this is a oscillation of gene expression. We screened transcription factors that are involved in the stress response and identified the two transcription factors, STAT2 and the ELT3. They are more enriched in the nucleide molting stage. And knocking down both transcription factors suppress the upregulation of VHA gene expression at the molt. So our work suggests that during molting, detachment of old cuticle from the underlying epidermis disrupt the attachment structure, and this structure damage can serve as a signal to trigger lysosome activation, including formation of tubular lysosome structures and increase of lysosome acidity and degradation activity. When lysosome is activated, it can facilitate degradation of all the cuticle components such as collagen, and the resulting collabites are used to build a new cuticle and therefore facilitate the cuticle repairment during molting. We have identified two transcription factors that STAT2 and the ELT3 that mediate lysosome activation by upregulating the VHA gene expression. Interestingly, STAT2 is known as a signal activated transcription factor that's associated with the apical HT component MAL4, so it can detect the structure damage occurred during molting, while the ELT3 is a tissue-specific, hypodermis-specific transcription factor. The combined action of these two transcription factors achieves a spatial and temporal regulation of lysosomes. And in addition to tubules that forms during molting stage at the level development, we found that many tubules appears when worms age. Here, worms, the average lifespan of worms is 20 days, so we count worms by days. Day one is the worm just entered the adult stage. We can see mostly their vesicular lysosomes, and gradually we see appearance of tubules, and they become more abundant in the enrichment at the later days. And under EM, we can see these tubular structures, but seems with very small content inside. And in addition to these morphological changes, we found that the lysosome acidity, motility, and the degradation activity all declined during aging. Here we use a lysosensor, and the ratio of lysosensor and the lysotracker to indicate the acidity of lysosome, and we can see a decline of the acidity during aging. We examined 85 lysosome expression of 85 lysosome genes, and we found half of them exhibit obviously downregulated expression at day 5 versus day 1. And this includes two major types of lysosome genes, VATPase and caspasins. The insulin and IGF-1 like pathway regulate lifespan and the DAR formation, which is a development stage in worms. The DAF2 encodes the insulin receptor. The dysfunction of DAF2 reduce the insulin signaling, and this can relieve the suppression of DAF16 by the insulin signaling, and therefore promote lifespan extension. So DAF2 loss function is a famous long-lived worms. The lifespan of the DAF2 is twice as long as in wild type. And interestingly, we found these long-lived worms contain the lysosome pattern in these long-lived DAF2 is quite different with from wild type, especially during the aging process. We see the DAF2 mutant can maintain their vesicular lysosomes and their EM. We also see these dense small vesicles, which suggest in addition, which is very different from the long tubes that we observed in wild type at the same age. And also, we see that the lysosome gene expression is obviously upregulated in DAF2, especially the caspsin and the non-proteinase hydrolases. And in addition to DAF2, which belongs to the insulin and the IGF-1 pathway, we found another two longevity regulated pathway. One is a dairy restriction pathway, and the other is a mitochondrial pathway. These are all evolutionarily conserved longevity pathway, also involved in regulating lysosome activity. Well, the lysosome function is important for the lifespan extension of these three pathways. In support of that, we have identified numerous lysosome defective mutant from our screen, and many of these mutants are short-lived, indicating that the lysosome function is important for longevity regulation. And we have identified two transcription factors, the FOXO transcription factor DAF16 and the NERV2 transcription factor SCN1. They act downstream of this longevity pathway to upregulate cathepsin and the VATPase expression, and therefore achieve the regulation of lysosomes by this pathway. So now we have seen two different types of lysosome tubules. One forms during larval development. These tubular lysosomes are acidified. They have increased degradative activities, so we think we call them degradative lysosomes. While we also see gradual appearance and the enrichment of non-acidic and also with a low activity, with low degradation activity during aging. So now we wonder how these dynamic changes of lysosomes are achieved. Remember that we have another type of lemma mutant, the lemma T mutant, which accumulate extensive tubular lysosomes. We have from this type of mutant, for example, the lemma 2 mutant contains more lysosome tubules from embryonic larvae to adult stages and in multiple tissues. We cloned these genes, we cloned the gene affected in this lemma 2 mutant, and this encodes conserved heat repeat protein called HPO27 in worms. And this HPO27 protein contains 37 heat repeats, and this heat repeat is named after four founding member, Huntingtin elongation factor 3, the A subunit of PP2, and the TOR1, all very famous proteins. And by using a Rosetta fold developed by Baker Lab, this heat repeat HPO7 appears to fold into a hook-like superhelical structure containing this containing the 37 heat repeat. The heat repeat, each heat repeat actually fold into a antiparallel alpha helix, and this 37 have a helix stack further into a superhelical structure. The HPO7 is well conserved through evolution. The human protein is called MRH1, and these two proteins exhibit a very similar tertiary structure. We identified this protein from the morphological screen. It contains many tubules and forming a tubular structure. Surprisingly, we found this tubular network cannot sustain and eventually lysosome collapse in these HPO27 mutants. And this lysosome causing the dispersion of the releasing of the lysosome hydrolysis here is the RNC2, the endorribonuclease, while the dispersion of SCAP3, which is a lysosome membrane protein. And this lysosome collapse phenotype occurs specifically in the epidermis, the big essential cells that I just introduced to you. And this phenotype increased obviously with age. So this data suggests that the tubular suggests that loss of HPO27 may cause structural damage of lysosomes. So to further examine that, we perform an EM analysis. First, at the day one, when 80% of the mutants still contain many tubular lysosomes, and the EM, indeed, we can see tubular structures of lysosomes, while at the day three, when 80% of the worms has no obvious lysosome fluorescence of lysosome reporters in HEPA7. And at this stage, when we do EM, we can hardly see any lysosomal structures, and the remaining ones seems to contain membrane walls and some cytosolic contents wrapping by the ER membrane because we saw ribosomes on it. And also, we see pieces of ER and fragmented mitochondria. So this data confirm that loss of HPO27 disrupt lysosome structures, and also and this lysosomal defect may lead to the secondary phenotype of ER and the mitochondria. And consistent with defects in membrane integrity of lysosomes, we see these mutants impairs lysosome cargo degradation with it accumulates high levels of GAP-LGG1, which is the LC3 homolog, and also have many apoptotic cells, indicating that autophagic and phagocytic cargo degradation is impaired. This mutant has obvious developmental defects. About half of the worms cannot survive during larval development. And this, when we look closely to these arrested larvae, we can see that most of them are enclosed within the L1 cuticle, while the others show obviously cuticle shedding defect, indicating that the HPO27 function is important for cuticle replacement, which is essential for larval development. And consistent to that, when we examine a knock-in reporter of HPO27, we can see that the HPO7 level is elevated obviously during molting. And also, in addition to the developmental defect, this mutant lives significantly shorter than wild type, and also it reduced the lifespan of the three long-lived mutant, including the DEF2 mutant, which is the insulin IGF pathway, and also the E2 and ISP1 mutant that belongs to the dairy restriction and the mitochondria pathway. And the yellow arrow indicating the lifespan of the long-lived worms, while the red one indicates the double mutant, which has lost the function of HPO27. So this data indicate that the HPO7 mutant affects longevity. So we have seen that this HPO7 mutant contain many lysosome tubules, and eventually tubular network cannot sustain, causing a severe defect in lysosome membrane integrity and the function, leading to defects in both development and the longevity. So we wonder why this mutant contains so many lysosome tubules, and to do that, we look carefully the tubule dynamics in both wild type and this mutant. We can see that the tubules, we follow the tubule formation, fission, as shown here, and also fusion and the retraction, the four different behavior of these tubules, and quantified different behaviors. And we found obviously that this HPO27 mutant has significantly reduced the fission events, while the fusion events is increased, but especially the fission between lysosome tubules, while the retraction events does not change obviously. On the other hand, in wild-type worms at both molting and the adult stage, we can see lysosome tubules. But when we overexpress SPO27, we can see it reduce significantly the tubule abundance. And this data tells us that these genes is probably involved in lysosome fission process, because loss of its function causes reduce the fission, while increase the expression of this protein can reduce the tubule abundance. We then generated the knock-in reporter of HPO27. This is a red one, and we can see it is associated with lysosome and enriched at specific foci on lysosomes. We then followed the dynamics of HPO27 using a super resolution time lapse. We can see that the HPO27 is enriched at the fission site and to mediate the fission of lysosomal tubules. Because HPO7 is a heat repeat protein that contains 37 heat motifs without any other domain, including transmembrane span. So we wonder how is HPO27 is recruited to lysosome. We first checked the whether Rep7 plays a role because it is the most important rep GTPs on late endosome and lysosome. And indeed, we can see that here is in the wild type HPO27, GAP forms a foci on lysosome, which is marked by the New-Quan cherry. And when we overexpress GTP-locked Rep7, we can see more HPO27 foci on lysosome, while when we express a GDP-locked inactive Rep7, it totally disrupts HPO27 foci. We then examine the protein interaction by an IP experiment. And we can see that the purified HPO27 interact specifically with a GTP bound Rep7, but not the GDP bound inactive Rep7, suggesting that the HPO27 is recruited to lysosome as a Rep7 effector. And then we examined the MRH1 localization and dynamics in COL7 cells. We can see that the GAP MRH1 associated with the lysosome labeled by Lamp1 cherry on both the vesicular and the tubular structures. And we follow it by, again, using a super resolution time lapse, and we saw very similar dynamics of MRH1. It is enriched at the fission site, and then we can see the fission of lysosomal tubules underneath the MRH1 foci. And also, like HPO27, our data show that MRH1 is recruited to lysosome as a Rep7 effector. And we generated MRH1 KO cells and found that the tubule, the number and the length of lysosome tubules all increased obviously compared with control cells, while the fission events reduced significantly, indicating that MRH1 also plays a very important role in lysosome tubule fission. Our data suggests that in worms and in COL7 cells, HPO27 and its human homolog MRH1 are important for tubule fission, and they enriched on fission site. While we can see the membrane fission underneath the HPO7 or MRH1 foci. So next, we want to know whether these two proteins indeed possess fission activity, or it is recruited, or is function as a scaffold to recruit other proteins that can mediate the membrane fission. And to test that, we adopted in vitro fission assay developed by Thomas Picardi's lab. In this assay, supported membrane tubes called SMERS are generated in vitro, and then we incubate the recombinant protein of HPO27 or MRH1 with these tubules, and then follow the tubule fission under confocal microscope. We first did a membrane dot spot assay and found that HPO27 bound to PA very well. So we code PA on SMERS. And the movie never starts from the beginning. So when we incubate HPO27 protein, we can see this protein can form bright foci on the SMERS, which is labeled by red, and we can see a membrane constriction and fission at multiple places, suggesting that the HPO27 indeed can cause membrane fission directly. And similar, we can see that the MRH1 protein can form bright foci along these SMERS, and to mediate the tubule fission and constriction and the fission. So this data tells us that these two proteins persist the membrane fission activity. Because we see a bright foci of both HPO7 and March1 that can form along these supported membrane tubes. So we suspect that these two proteins can form oligomers. We did negative EMs, negative staining EM, and indeed, in solution, we can see HPO27 and March1 mainly form monomer and dimer, and this monomer structure is very similar to the predicted structure, the hook-like structure. In very rare case, we can see HPO27 form different types of polymers. And then we incubate HPO27 protein with nanotube. And in this case, we can see HPO7 can form polymer structure around the tubes. Because we saw both opened and closed ring structure of the polymer, we guess that this protein forms a head-to-tail linked polymer. To test that, we use tripartite split GAP assay. In this assay, we fuse two GAP10 or 11, the small fragment, to the terminal of the protein, while express the big one in the cytosol. And we so when this when the complex formed, we can see reconstituted GAP fluorescence. We express this cassette in worms, and we saw that only when we fuse two small fragments to either C or N terminal of HPO27, we can see reconstituted GAP fluorescence that overlapped with endogenous HPO27. But if we fuse both fragment both at N terminal or C terminal, no reconstituted GAP fluorescence can be observed, indicating that indeed it is a head-to-tail connection. And similarly, we can see a head-to-tail assemble of MRH1 in worms as well. And then we wondered how this head-to-tail is formed. So we removed the terminal heat repeat from HPO27, and we did a tripartite split GAP assay. And in this case, no fluorescence can be observed, even we fuse the small fragment to N or C terminal. And also, we managed to purify the delta C HPO27 protein, and it can only form monomer in solution, suggesting that the terminal alpha helical module is important for the self-assemble of HPO27. And this HPO27 delta C still bind to PA, but it failed to assemble on SMERS in vitro or assemble on lysosomes in C. elegans. Also, they have this truncation version of HPO27 lost fission activity in vitro or in worms, indicating that self-assembling is very important for lysosome association of HPO27. So our data suggests that HPO7 and March1 is a lysosome fission factor. It is recruited to lysosome as a REP7 effector and enriched at the fission site forms it assembles in a head-to-tail version of a polymer to mediate membrane constriction and to mediate the constriction and the fission of lysosomal membranes. Loss of HPO27 or MR20 deficiency impairs lysosome fission, causing accumulation of lysosome tubules, and eventually impairs lysosome structure integrity and the function. In worms, it causes defects in development and longevity. And as you probably will realize that REP7, although REP7 recruits HPO27 or MR22 lysosomes, but these two proteins are further enriched at the fission site. So we guess that phospholipids or membrane curvature may play a role in the final targeting of the protein to the fission site. And the other interesting point is that in the in vitro assay, HPO7 and MRH1 mediate the membrane fission without direct use of energy molecules such as GAP or such as ATP or GTP. And neither of the protein is ATPase or GTPase, indicating that they represent a new type of fission factor. So we are very interested in understanding how this molecule mediates membrane fission. I hope that I can convince you that worms can serve as a powerful and effective model for the lysosome study. And in addition to the not only at the cellular level, but more importantly in the context of development, aging, and the stress condition. In addition to lysosome morphology and dynamics that I presented to you today, we are also interested in understanding lysosome biogenesis activity and integrity. We hope that we can dissect lysosome homeostasis using C. elegans as a multicellular model and understand how it is regulated at the cellular level and under the condition of and the different contexts, including development, stress response, and the aging. So finally, I want to thank my students. Many students actually contributed to our work, from phagocytosis to lysosome. So to the stories that I present today, Bing and Hongwei did that one work. Ray characterized the lysosome function during molting, and Yanan did the work of lysosome function, the morphological change of lysosome and regulation during aging. And the HPO7 and MRH1 study is a project with a long history in my lab. It's a teamwork within my lab and also with my collaborator. When we initiated the project, identified HPO27 mutant and did the initial characterization, while Le Tao did most of the work I present today, and Shilu did genetics and cell biology analysis. And also, I'm very grateful to my collaboration, the collaboration between my lab and Weifeng's lab at IBP, Shenzhen and Wenjuan from Feng Lab contribute to the protein purification and the characterization, as well as the negative staining EM, while Mei Jiao from Chonglin Yang's lab at the Yunnan University did EM analysis in worms. Also, thanks to my collaborators, Chonglin at the Yunnan University, we collaborated in many different projects, and Huimin Zhang from Suzhou University gave us a lot of help in the molting project. I'm very grateful to my colleagues at IBP, and we have a really good cell biology group. Weifeng, I just mentioned, we collaborate on HPOs and MRH1 project. I learned a lot from him. And my cell biology groups in the IVP, Junjie, Dong, and Hong. And our worm strain is from Tokyo Women's Medical University and the C. elegans Genetic Center. And also thanks for all the funding agents that support our research. And I also want to thank my previous institute, NIPS and IBP. Okay, so finally, I want to thank very much for all the audience staying with me for so long. Thank you, and I'm happy to answer any questions.
Thank you, Xiaochen. This is really beautiful talk. And so, so for the audience, please type in your questions to the chat box, and I'll read it to Xiaochen. But maybe I'll begin with the questions that I have. And so I have two questions. One is on the first part of your talk, when you describe this immunoacid acid transporter mutants, which has this enlarged lysosome phenotypes, and try to link that to your second part of your talk. Do you know what's causing that lysosomes become so big? Is it because there's more fusions or less fusions, or there's more lipid transfer so that you can actually keep the building up the membranes there?
Yeah, we actually do not know exactly the reason of the lysosome enlargement in different cases. The common feature of this mutant or the lysosome, enlarged lysosome in this mutant are the accumulation of different types of cargos. It seems not the swollen of lysosome. And another point is that we did not look very carefully about in terms of fission and fusion in different mutants. So I cannot say that whether fusion is increased or fission is decreased. Yeah, we should look at it. But one common thing is that these enlarged mutant do not have many lysosome tubules, or I would say they are defective in tubule formation. So you have to have a normal lysosome to form tubules. Yeah, but we don't know we don't know whether fusion is increased in this mutant, but we do see accumulation of many cargos.
The second question I have is about the HPO27. I wonder whether the level of HPO27 is regulated, particularly in the context of lysosome damage, because you could imagine that maybe when lysosome is damaged, and you may want to cut off certain part of the lysosome that is damaged, and so that they can actually go to lysophagy, for example.
We don't see upregulation of HPO27 in our mutant that have integrity defects. While in worms, we still do not have a way to induce damage. But we do have mutants that are defecting in integrity and therefore accumulate damage lysosome. And in this case, we do not see upregulation of HPO27 or accumulation of HPO27 specifically at the damage site. Yeah, but we haven't. We can look. We actually can look at that in mammalian cells because we can see whether MR1 is recruited to lysosome in response to damage. Yeah, we haven't looked at that. Yeah.
All right, so there are a couple of questions from the audience. I'll read it. The first one, you may ask, and what is the functions of the tubular lysosomes?
Yeah, this is a good question. Well, the short answer is we don't fully understand the function of tubular lysosomes, but we have several guesses. For example, during molting stage, we do see that the tubular lysosome formed, and we actually did a FRAP analysis. We know that the tubular structures are connected inside. So if we bleach, we quench the fluorescence at one place is somewhere in the tubule, and we can see it recovered quickly. But if we bleach the fluorescence in a vesicular lysosome, it cannot be recovered very well. So one thing we think a tubular lysosome is a facilitated, or actually is a facilitated cargo distribution or catabolized distribution. And the other thing is we have the epidermal cells that we use is very big, as I have mentioned. It covers the whole mid-body of the worms. So we think the formation of a tubular lysosome actually can increase the capacity of lysosomes for both degradation and the cargo and the catabolized recycling, and therefore increase the overall degradation or recycle capacity of lysosome system. This is our theory at this time. But we do need other or more direct evidence to really demonstrate the functionality of tubular lysosome.
So another related question is, are the tubulin lysosomes generally more aged than the ellipsoid ones? And what is the evidence for the assumptions that the lysosomal aging processes are synchronized with worm aging?
Uh, are the lysosome generally more aged than the ellipsoid ones? For assumption that some aging process are synchronized with the, I don't think we can answer that. Yeah, I actually do not understand this very clearly.
I guess the question is whether you see more tubular lysosomes during aging, because you show that during aging, the lysosome pH is changed, right? Or I remember it was one slide showing that. And do you see the
Right, so
morphology change as well?
right, so I think we see the age associated lysosomal change, including increased abundance of lysosome tubules and decline in lysosome acidity and the degradation activity. So they occur, I would say, seems associated with each other, but I couldn't say that which one is cause, which one is consequence, or none of them actually is the cause.
Right. Related to that is next question is whether HPO27 from Fenghua and HPO27 level is changed during aging.
Yes, we do see a reduction of HPO27 level during aging.
A reduction.
Yeah, a reduction of the HPO27 level during aging.
Yeah, Fenghua also asked this questions, I think probably pop up in everybody's minds. And what is the energy source for HPO27 mediated
Yes,
aging?
we want to know. Yeah, we actually are very curious about that, but I don't know at this point. I have some guesses. It is clear that in the in vitro assay, it does not need a GTP or ATP because the system only have a recombinant protein and the supporting membrane tube. And we see a concentration and fission occurs that way. But in vivo, we don't know how the cellular energy is used. And my guess is because the protein is a special, the heat repeat protein actually is a special. The most distinctive feature is the flexibility of this protein because the heat motif is an anti-para. Each heat repeat is anti-para alpha helix. And the whole molecule is like a spring. So there are some studies that can fix one side of the protein, and then you can use a force to extend the protein. And then when the force is losing, the protein actually resume its original conformation. So I guess the energy may be buried within the conformational change of the protein. When the polymer is assembled, or it may be there is a disassemble of the polymer, and the energy may be buried within the conformational change of the protein. But we need to prove that, or at the first step is we need to see the structure of this polymer, and then find a way to study how it is assembled and whether it is disassembled, and if so, how the disassembling occurs and contributed to the membrane fission.
Xian Han says, great talk, and he's wondering, lysosomes of different cell type must be different in size and shape and functions. Can you comment on the change of lysosomes dynamics of different cell types?
Yeah, we do. Yeah, we do see lysosome have very different shape and behavior in different cell types. So we use this hypodermal cell to do the studies. One thing is because one reason is because it's big, and the other is it has very abundant lysosome dynamics. But in other cell types, for example, in the intestine, or in the muscle body wall muscle cells, the lysosome mostly are still vesicular lysosomes, and dynamic change do not occur. That I would say we don't see very many dynamic changes of a lysosome, and also lysosome, one feature of lysosome is the heterogeneity of lysosome, including the morphology, activity, acidity, things like that. So I think we still do not fully understand, for example, in worm intestine, what are the lysosome and whether they are the same or not. And in terms of lysosome dynamics, we see different behaviors in different cell types. And also we actually are curious of lysosome in neurons, but they are too small, so we still do not know whether neuronal lysosome change their shapes or activities. But from as far as we know, or as far as the different cell types that we examine, lysosome behave differently, and their function or their activity change differently at different stages.
So, Hongyan, I'll ask two questions. I think these are both also very good questions. The first one is why the collapse of lysosome only happens in HP1, HYP7 cells, but not in the neighbor cells.
Yeah. One thing we are wondering is it's because of the hyper7 cells has very active lysosome activity, or what I have to rephrase that, is we have several cases. For example, our guess is like the tubular lysosome structure cannot sustain if lysosome continue to import cargo into it, or export catabolites from it. In other words, if lysosome fission is impaired, such as in the HPO7 mutant, if you stop any activity of lysosome, the membrane integrity is probably preserved. However, if you continue to input the cargo into these tubular structures, and also degradation activity continue to occur, and the catabolites is export as well, and then eventually the structure cannot be sustained. We have several observations. For example, if we block a tubule, if we block a cargo input, we actually can suppress some of the collapse of lysosome tubules, partially suppress the collapse phenotype. But if we increase the cargo input, for example, starve the worms, in this case, autophagy activity is accelerated, and a lot of autophagic cargo are delivered to lysosome. We can accelerate, we can increase actually, or increase the collapse of lysosomal structures. So we think it's lysosome, it's probably a threshold. Well, the continued activity of lysosome cargo input and the catabolite export will eventually disrupt the structure or the network.
So it's also asking whether those tubular lysosomes are more acidic or less acidic.
I cannot, yeah, I don't know. Yeah, I'll say that we have not carefully compared the acidity of tubular lysosomes versus vesicular lysosomes, because we still do not have a very good way to measure that accurately in the case of wild type, when fission is normal. Yeah, we haven't done that under exactly the same condition to compare vesicular and the tubular, but to measure acidity by, for example, lysotracker staining, that normally you will see more stronger fluorescence in the vesicular lysosome, but I think it's because of the shape. of lysosome, but not exactly the acidity in different shapes.
I think for the sake of time, we have to stop here. I'm sorry that there's still questions there, and maybe I can write to Xiaochen later on to further discuss this.
Sorry that
I know
yeah,
it's
I
a
talked too many
fascinating talk,
times.
and yeah, there are a lot of questions, I'm sure, from the audience. And thank everyone for being with us so late, and also thank Xiaochen for this wonderful talk. And all right, so I'll see you all in a month.
Thank you, Yihong. Thank you, everyone, for staying with me for so long. Thanks.
Thank you. Bye-bye.
Bye-bye.